everybody. I'm Cameron Lamoureux. I am a fifth year PhD student in the Systems Biology Research Group here at UCSD. I'm interested in studying large-scale data sets relating to genomics or transcriptomics in the model organism E. coli. I find it really fascinating to apply large-scale analytical techniques, machine learning, statistical methods to biologically relevant data as part of broader efforts to convert big data to knowledge. So this study is all about data. How can we assemble it, control its quality, and most importantly, learn from it? In the last 10 years, our lab has generated over 1,000 data points measuring expression in the model bacteria E. coli. Expression refers to the set of genes that are active at any given time in the bacteria. As the bacteria is exposed to different conditions, there could be different sugars to munch on or different stressful environments such as high temperature or acidity, the set of genes that are active, or the expression, will change to reflect the needs of that particular situation. These changes, in turn, are mediated by a control network. So we have these 1,000 measurements of expression. Can we use machine learning to extract the organization of their control network just by looking at that raw expression data without using any prior knowledge of the network? It turns out the answer to this question is yes, we can. And this is something that our group has already demonstrated and pioneered by extracting 92 imodulons, that's the name we give to these recovered control network signals, from a 278 sample data set. But what happens when we scale our data set to over 1,000 samples? Do we learn more? Do our analysis methods even still work? Does our understanding of the control network apply to new data that we haven't generated? And what if we want to scale the data even further? Do we continue to get a similar amount of control network discovery, or are there diminishing returns? All of these questions motivated deep analysis of our large-scale expression data set, as we are deeply interested in converting big data into biological knowledge. First of all, the raw expression data itself is quite useful. At this 1,000 sample scale, we are able to identify broad patterns in gene expression in a variety of different conditions. And we call this 1,000 sample expression data set precise 1K. We do indeed find that we can extract more knowledge of the control network when we have more data. In total, our machine learning algorithm recovers 201 imodulons that account for about 86% of the known control network activity. That's up from under 50% in our smaller scale previous study. We are also able to find previously unknown signals that go beyond the current annotation of the control network. So we're not just recovering signals, we're discovering signals. We also explore an even larger scale data set by adding about 1,700 quality controlled, publicly available samples that we didn't generate to our precise 1K data set. We do find some diminishing returns in terms of the recovery of the control network at this scale, but importantly, we demonstrate that our analysis methods not only scale to this 1,000 sample data set size, but all the way up to a nearly 3,000 sample data set. Perhaps most importantly, we demonstrate that we can use our recovered control network signals, these imodulons, to explain data that we didn't actually generate. Our imodulons concisely but effectively capture the network signals active during transition from an oxygen-poor to an oxygen-rich environment, which is a critical transition to study for this gut-dwelling bacteria. This study demonstrates the recovery of expression control network signals at extremely large scale. We show that our machine learning-based method is scalable and robust, and we demonstrate new biological knowledge. Most importantly, though, we provide a framework for analysis and contextualization of new expression data. Because precise 1K is large and contains many different conditions, we capture a broad spectrum of control network signals in different levels of activity. Thus, when new data is analyzed against these signals, The 1,000 data points of Precise 1K function as a massive set of controls, a background upon which new data can be overlaid and understood. We believe that this capability represents a fundamental shift in how expression data in E. coli can be analyzed. Two aspects of this study were particularly challenging. The first was dealing with the large scale, processing all of the raw data, running quality control, running our machine learning analysis workflow. All of these steps had to be modified somewhat to generate results. We benefited greatly from cloud computing and high-performance computing infrastructure for this study. The second big challenge was perhaps a happy one, understanding exactly what to analyze. This data set is so large and so rich that we had so many different ideas for ways to investigate it. And we're sure that though we've detailed many of these in our paper, we've still only just scratched the surface. From the expression data itself to the imodulons extracted from it, we do believe that a huge additional opportunity for data mining and data analysis remains. One key, somewhat unexpected result was the significant diminishing returns of regulatory network discovery upon adding 1,700 high-quality, publicly available transcriptomes. We only nudged the needle from 86 to 88% of the known regulatory network recovered by imodulons, despite more than doubling the size of the dataset. We believe this is ultimately a result of the fact that the remaining minority of the network is responding to very specific signals that were simply not activated by any of the conditions available to us. If part of the regulatory network is responsible for activating metabolism of an exotic substrate, but that exotic substrate is never provided to the organism, that corner of the network will remain dormant. No change to any gene expression levels will be affected, and since our machine learning methods depend on variation in expression to pick out signals, they remain blind to these unactivated cases. However, the publicly available data highlights a very positive result, which is the sufficiency of the existing imodulons to explain complex regulatory network responses during critical biological dynamics. As I mentioned earlier, we were able to broadly deconstruct the regulatory network activity of the anaerobic to aerobic transition, contextualizing the activity level of regulatory network elements against all Precise 1K samples. The resulting analysis is a critical blueprint for similar data mining efforts in other publicly available data or any new data that is generated over time for this critical model organism.